Good morning.
Good morning, Liz, and welcome to everybody this morning. It's lovely seeing you all, and thank you for turning up so early to our meeting. We're very lucky today because we've got two fantastic speakers, and we've named this session Beyond the Numbers because statisticians, as you mostly know, are amazing people. They not only know mathematics, but they actually can advise us very much more generally in the farm industry. I've had an amazing time. I was just saying to the guys before we started today that my first statistician was a lady called Karen Facey, and she helped me with the MHRA assessments incredibly. And I learned at a young age, certainly quite young then, that statisticians are very much thinkers. They can actually look beyond the numbers. They look at the totality of what you're trying to achieve, and they basically show you and can advise you on how to get to where you need to be. The probability of having two amazing speakers this morning is probably less than 0.005. But we have these two gentlemen who very kindly agreed to speak for us. They both are extreme leaders in their field. They have both had incredible experience across a whole range of clinical views in terms of phase 1 through to phase 4. And they've advised a lot of bodies out there. And some of the concepts you're going to be hearing this morning are known to you, and some are probably less known to you. And it's my great pleasure to have them speak with us. Our first speaker this morning is a gentleman called Dr. Nelson Kinnersley. And Nelson is extremely well known in the industry. He has got a PhD in biostatistics from Imperial College London. He's got over 30 years experience, although you wouldn't guess it by looking at him, as a biostatistician in a wide variety of areas in the farm industry. He's got experience in oncology, neurosciences, virology, and after many years at Roche, he founded Octa Consulting Services in 2020. He's got his main interest in strategic input into drug development, as I think most of us have, and is the entry into human first-time man studies all the way up to the licensing. And he's very passionate about the flexibility in study designs, which you'll hear about this morning. And he really wants to maintain the vigor and the way that we look at these studies and the way that we adapt them. He also serves as an independent statistician on data safety monitoring boards, which is an incredibly tough job. So Nelson, I'd like to very much welcome you, and thank you very much for your talk this morning. Thank you.
Well, thank you very much indeed. And I think, are we going to do poll 1 before we get into the talk?
We will do poll 1. So Anna, is that okay if we can run the poll? So here we go. Which attributes come to your mind when you think about statisticians? Now, Nelson, this is a very interesting one. This is something I might have to admit that we put together with the agreement of the two gentlemen. It's not my thinking, it's their thinking. So do you think of statisticians as being nerdy, difficult to understand, smart, easy to work with, or strategic? Now, this is silly really in some respects, because I think they're probably all of them in many ways, apart from, in my view, nerdy. But let's see what everybody thinks. So push your buttons and let's see what the result is. Give you a moment or two with that. Right. Has everybody submitted, Anna?
Yes, I'll share the results.
Okay, fantastic. Oh, what do you think, Nelson? You can comment, not me.
That's very interesting. And I think that, you know, in some ways, we've got a bit of a self-selected audience because you, and I say you know, Liz, Anna, Malcolm, you put a lot of work into preparing for this. And so I hope that the people who have signed up for this are going to get something out of it. I can't help be drawn to the nerdy statistic in there, but yes, of course, you know, many of us like to work in multiple areas here, strategic, and I think quite an often criticism is difficult to understand, and you know, communication is something that I think all of us try to do our best, but we don't always succeed in that. So yeah, that's some nice data.
Great. Okay, Nelson, over to you. Thank you.
Cool, thank you very much. Well, just to orient everybody, I'm going to drive the slides that Anna started off sharing. And I almost want to give up at this point because Malcolm has given an introduction for Alexander and I that I'm just wondering how either of us can live up to. But you know, that's one of the tasks that we're here today, is to talk a little bit about our passions. I've got a number of slides which I'll walk through. There is no algebra in this, which, you know, given the early hour, hopefully that will go down well. But there's quite a lot of screenshots, and I really want to try and encourage a dialogue when we get to the Q&A at the end. So let's jump in. So just in terms of taking a little bit of a step back, you know, the title of this talk is about trial design. And you know, one of the questions is, isn't everything already known? I think another way of describing this is, well, how often do new study designs get published? And what I'm showing you here, particularly circled in red, are just 10 examples taken from publications of new study designs that have been published since the start of this year. Now, maybe that's no surprise to you, and you're used to seeing this, but I must say that in some of my experience, there's a number of people believe that, well, all the study designs have been put out there, and yes, there may be one or two here and there, but they've perhaps been quite surprised that the number of new designs comes out very, very frequently indeed. And so, you know, what potentially are the implications of having so many designs published frequently? I'm just trying to click through. Here we go. Thank you. So I think one of the things that I encourage people to do, and I'm sure many of you do this already, is before you go into your discussions, your clinical development planning, evaluate what your available designs are. That can be done maybe on a theoretical basis, maybe that can be done on a more pragmatic basis, where we have the experience of people who've published trials already. I've got a little bit of a delay there, Anna, just coming through. So if you can just bear with me, I've tried to click backwards on this. And we may end up just reverting to you controlling it, Anna, if there we go, we're back. Something which I know statisticians get deeply involved in are performing simulations for the specific situation. I think if there's, you know, a couple of messages I want you guys to take away today, is that really there isn't one design that can suit every single situation, every molecule, every disease area. And simulations can be quite a powerful tool to take the context that you and your teams are operating in. And often statisticians can help inform other team members of what the trade-offs are. And we'll talk through some of those things hopefully today as well. And the other thing I think, being realistic, is if designs are coming out very frequently, just be prepared that, you know, new designs will appear after you've selected your trial design. You know, in some ways, is it like buying the next, you know, your latest mobile phone? There's always one that's going to come around afterwards, but how much of an incremental improvement is perhaps an open question in that sense there. So the objective for today is to remind you of the classical approaches to early phase designs in oncology, and then to show some examples of some alternative designs for phase 1-2. And one of the things that I want to be able to try and do is at least pique your curiosity. And I'll be showing you screenshots of the MD Anderson's online apps. And I personally believe they've built some very, very nice features into this, and will help both statisticians and non-statisticians navigate some of these available, but, you know, really encourage you to experiment in your own time. So let's jump into the classical approach, or the 3+3 in oncology, and let's talk about what this is here. So generally, as I'm sure all of you who work in the oncology, but maybe some of you outside of it are somewhat familiar, that what we try and do here is identifying doses that lead to unacceptable toxicity. So it really is about safety, and we'll declare the maximum tolerated dose, or MTD, somewhere below. And the rationale really is that increased toxicity should lead to increased efficacy. That's one of the rationales of the original 3+3. And how's that done? I'm sure you've seen images and schematics like this before, but we identify a starting dose, usually very low, and there's various regulatory and other guidelines for how to start, choose your starting dose. We need to pre-specify which adverse events form the DLTs, and then we'll classically, at least with the 3+3, we enroll groups of 3, or we extend that up to 6. And the stopping criteria are when we have either 2 of the 3 in a cohort, or 2 out of 6. This is, if you like, the classical 3+3. And what are some of the pros? It's heavily and widely used. There are more than 90% of oncology phase 1 trials were documented in a survey in the mid-1990s and onwards, and it really has had a very widespread adoption in there. So I think we've just gone through back for that. What are some of the cons? It's been well documented in the statistical literature and maybe slightly wider, but it's a design that's poor at identifying the MTD. So, if we look at the top of this slide, the aim is to declare the MTD, and yet it's poor at actually identifying it. And that mechanism of action for cytotoxics really was the original driver for this. I'm sure much of this is well known to this audience, but I'm choosing this to ground some of the other designs that we'll go on to talk about. So let's see an example of a 3+3 in a simulated world. If we look at that schematic, we see that approximately, we can say the target toxicity is 25% in there. So let's do a simulation. And in the first scenario that I want to simulate, we've got 4 available doses, and the toxicity for dose 1, 2, 3, and 4 are those percentages there. So we see 5%, 10%, 20, and 60, etc. Whereas in scenario 2, the dose 3 is bang on the 25%. And so we can run these simulations and study, well, how often is the MTD correctly identified round about dose 3, certainly for scenario 2 and 1. And as we see in this simulation here, dose 3 is selected as the MTD 55.1% of the time. In other words, 44.9% of the time, that MTD is estimated incorrectly. So that's not exactly promising in that regard here. One of the other features, now we look at scenario 2. So this is where we know we've simulated this thousands of studies in which dose 3 has exactly 25% target toxicity. And yet the 3+3 only picks that up 42.2% of the time. I think many of us would argue that that's poor. One of the other features I'll just show you, this is half of the patients in the trial receive very low doses with toxicity less than 10%. So this is the percentage of patients treated, 23.7, 30.1, that's just over half of the patients are receiving very low doses. So arguably that's not a super design in that sense. So what are the implications for teams who choose to run this design? Potentially taking suboptimal doses into expansion cohorts or later trials. And really, I think one of the things that you maybe think about is that while it might be tempting that your goal is minimize total sample size, I'd like to encourage people when they're thinking about designs that there may be some other criteria. How good is the design at selecting the right dose? So let's look at an alternative here, the continual reassessment method published in 1990. And what this does is it assumes a mathematical model can predict the probability of toxicity. You enroll patients and you continue to update that model. And then you stop enrolling once you've have sufficient precision. So here's some examples, worked examples taken from the reference at the bottom of the page there. You see there's these little dashes, or some people call them rug, at the bottom that shows you each time there's no toxicity. And at the top of cohort 4 here is a toxicity. Cohort 5, there's an extra rug dash down at the bottom. So you just go through here and the model updates itself with each toxicity or no toxicity. So after cohort 5, The best estimate of the MTD is 410 milligrams. As you can see, that's where the line, the toxicity line at 25% intersects with that model curve. And you just keep going, and you have more and more toxicities and non-toxicities until you've got a sufficiently precise estimate. So for example, 475 is your MTD after cohort 15. Absolutely want to acknowledge that many variations have been published since the original CRM from 1990, but there's not time to go into all of those today. One of the features that's been highlighted in some of the statistical and other literature is that you really do require a statistician at both design and conduct. Now, for people like Alexander and ourselves and a number of you on this call, great, you know, keeps us in work, but that's not always open to all sponsors. And this is arguably maybe one of the barriers to not a huge uptake in the past 30 years. So let's look at an alternative Boyne or Bayesian optimal interval design. It allows you to have flexible cohort size. As we know, 3+3 classically only has cohort size of 3 and 6. I would argue it's simple to design and conduct, and it still quantifies the uncertainty that's present when we're talking about estimating DLTs. I want to just try and give you an intuitive explanation of what Boyne is doing. You know, suppose this schematic with the colors in here represents the probability of a dose being toxic. If that probability is at the left-hand end, it's kind of 0, a dose, then intuitively we're in a very low dose, let's escalate. Then conversely, if the probability of a toxicity on the dose is very high, then intuitively we want to de-escalate. And then there's going to be some kind of sweet spot in between where what we do is we stay and we enroll more patients into the cohort. So the original authors, Liu and Wan, did some so-called fancy maths, and they showed us where those boundaries should lie depending on what target toxicity. So for some of the programs, you might have 30% target toxicity, and they show that those boundaries lie at 23.6 and 35.9. But you know, if you have a 25%, then you'll have different boundaries for where that sweet spot is in between and where you'll enroll extras. Something which has caused confusion, at least in some of the teams who I've been part of when discussing this, is that they've been concerned that the Boyne gives the answer and it must be followed by the DMC or a safety oversight. And I'll try and make and strive to tell people, no, the Boyne provides a recommendation. The fully empowered DMC, Safety Oversight Committee, are responsible for taking decisions. So let's very briefly do a worked example. I've chosen one here with 30% target toxicity. So there are those boundaries for the sweet spots in there. And if we are currently looking at 3 have been recruited, 0 out of 3, well, that's a probability of 0. So we'll escalate, we're on the left. 1 out of 3, as you can see, 0.33 is in that sweet spot. So we'll stay and we'll add more patients to this current cohort. 2 out of 3 is 66%, so we'll de-escalate. And finally, 3 out of 3 is 1. And this is a de-escalate and never use this dose again. It's the so-called eliminating this design in here. Just moving my mouse. If on the other hand, we've got 5, say, you can go through exactly the same process and work out what are the escalation and de-escalation rules. And so what that allows us to do is write into the protocol, even before the study started, what are the escalation and de-escalation rules. And so a table like this, or a schematic like this, can show, for example, I'll just pick the 3, the cohort size 3. If there is 0 DLTs, we escalate. If there's 1 DLT, we stay at this dose. 2, then we de-escalate. 3, we would de-escalate and never return to this dose. But all this can be pre-specified, and you do not need a statistician during study conduct. You can just read off. I've been asked questions about how widespread use is. I think it's fair to say it's increasing. I just did a search of Boyne the other day and came up with 67 hits. Admittedly, not all of those are exactly coming on Boyne using a Boyne design, but the vast majority that I scroll through. So yeah, just go on to clinicaltrials.gov and have a look in your area if you're interested to do that. But I also want to highlight that the FDA have reviewed this as a design and declared it fit for purpose. Now, I absolutely want to say that this does not mean the Boyne is the one and only design that the FDA accept, and the FDA are at pains to say that. However, if you're ever on Teams and they say, oh, we don't know what the regulators think, well, maybe the view of the FDA could help that discussion to at least say that they've declared it as a design that's fit for purpose. The footnote at the bottom, I want to just highlight this Boyne fits into a class of designs called model assisted. And there's something called the MTPI, or modified toxicity probability interval keyboard design. There are other versions of this. I'm just showing you for the sake of time today one of those where you do not necessarily need the statistician during the study, but we can help plan the study and write into the protocol. So at this point, what I want to do is just flash a bunch of screenshots at you and really encourage you after this, well, hopefully not during my talk and Q&A and Alexander's talk, but just go to the MD Anderson website, trialdesign.org, and they have developed a suite there that you can click on and you can start exploring some of these designs. Let's say I'm just going to click through, here's some phase 1 designs, and you click and then, oh yes, there's the Boyne, but there's a few other modifications and variations of it. You can generate your protocol schematics and tables from there as well. If you're not sure which version of Boyne you might want, perhaps you want to consider both safety and efficacy, then they provide some nice tools that walk you through some choice of study designs. Many of you, I'm sure, if not all of you, will be very familiar with the Simon's 2-stage. Again, just go and have a look at it. And if you've never really understood it, maybe the MD Anderson app can help you just take that little bit more of a deeper dive. One thing that I'm sure many of you have seen about the way that early phase designs and in fact review of drugs are going is around dose optimization. And in particular, the FDA announcement of their Project Optimus, which many of you may have attended webinars or various other readings about this. But I think it's fair to say that times are changing, and some of the experience that is very much in the early days, but it's coming through that the agency do want to see, in general, more characterization of dose toxicity and dose efficacy. And there's this well-documented example from last year where some small biotechs, in these words, spooked investors. Here's a list which is going to be available in the slides after the presentation today. Is really to encouraging, if you're not doing it already, is rather than thinking, well, what design am I used to doing? How can it meet the objectives? Think about what your objectives are and then get to study design. Now, I know many teams, many good teams have this as their practice already, but I've also been part of teams or overseeing teams where the design is picked first, and then the question comes, well, what are we trying to optimize? Is this about safety? Is it about efficacy? Is it activity? Is it about speed? Is it about learning about biomarkers? And of course, many people say, well, actually, I want all of these things and I want it tomorrow. Well, good luck with that. I would rather approach it, what are your key objectives? And identify that, talk as a team. This is not exhaustive, but it's some of the things that I certainly would, consider as part of working on development plans. So in summary, as we just finish up here, hopefully you've just had a very whistle-stop tour looking at some of the more classical, 3+3. We looked at model-based, so the CRM in particular, and then the model-assisted, Boyne and MTPI, and acknowledging that dose optimization is going to become increasingly important. So before I hand over, actually I think it's back to Malcolm to do a poll, what I'd like to think about is that I've at least started to pique some of your curiosity, encourage you to look at the MD Anderson website, talk to your statistical colleagues, maybe about some of the existing designs, maybe some of the new ones, some of the questions and things you've never thought about. And don't forget about simulations that really get to the point of, at least I would argue, there is no single design that works for every single context and every single question. And simulations can be a useful tool to help teams whittle down to a small number of designs, maybe even just one design that will then be good to go forward. So Malcolm, I think, is it back over to you?
It is, Nelson. Thank you very much indeed. That was absolutely amazing and an incredible overview. And thank you especially for that last section in terms of making statistics really, really interesting. And I suppose quite fun, actually. I will go on to that and really look through it because like many of us, we're asked about, which design, how we're going to do something, what sort of output. And quite often the output in terms of safety or efficacy is a question we're often asked as pharmaceutical physicians. And often we just say, we're not quite sure. So Nelson, thank you so much. That was really, really very, very interesting and fantastic. We're going to keep Q's and A's to the end generally, but some of you already putting the questions in. Thank you, Graham. I see you've already got one in. Thank you. So we're going to now do poll 2, Anna, is that right? Or we can come to the end of it. Okay, fantastic. Right, so we've got a second question here. When is the right time to start involving statisticians? When speaking about the challenge of the study we should address, when it's time to design the study, when it's time to think about analyses of the study, or when the data is available. So I'm sure we've all got views on that one. So please push your buttons and we'll see what the result is. I think I've got my own ideas about that one. Right, Anna, is everybody in?
Yeah, sure.
Great, let's have a look at the results. So, when speaking about the challenge of studies addressing, I would agree with that. When it's time to design the study, yep. When it's time to think about analyses, yeah, I think it's probably a bit late. When the data is available, yeah. Probably a little bit too late on, I'd have thought, the last one, but no, thank you very much indeed. It shows a range of ideas and thinking about way stats would work and the way that we design our studies. So that's great. Thank you very much indeed. Right, I'm going to close that down on my screen. So our second speaker is Dr. Alexander Schacht. Now, Alexander is, again, another amazing speaker and another guy who has got a huge amount of insight into this area. He studied mathematics originally, as I'm sure everybody does in stats. He's got a PhD in biostatistics of non-parametric statistics, which I'm sure I know very little about compared to most of you guys at the University of Göttingen. He's a statistician and executive vice president of launch and commercialization of data sciences at Veramed. He's authored more than 70 scientific papers, which is very impressive, and is regularly a speaker at international conferences. And he's got more than 20 years experience working as a statistician in healthcare, working primarily on the phase 2B/4 side of the industry, with some work on early phase as well. He's interested in a broad range of methodological areas with a focus on helping pharma and biotech companies make better data-driven decisions. And he's a very well-known podcaster in this area. And we're talking before when he's got many, many, many followers on his podcast, and he's got some very fancy kit as well, which makes me very jealous of him in terms of microphones and stuff. So, Alexander, welcome. Thank you very much for your talk, and it's over to you.
Thank you for the introduction. Yeah, my passion is really about making sure that there is the right evidence at the right time in the right format to make sound decisions for payers, physicians, and patients, because ultimately that is really what is needed. Next slide. So when we want to launch a product, Yeah, we do all the great stuff that Nelson talked about, you know, great phase 1, phase 2 work. We have the confirmatory studies we have, and now we have regulatory approval, and we want to lift off and get, you know, the patients this new therapy and make sure they benefit from it. But that's not how it works anymore. I was some years ago working on a new compound, and it happened exactly the following. You know, everybody worked like crazy on getting the regulatory approval. And then when the regulatory approval was there, the company wasn't able to get it to patients. Actually, they needed to delay bringing it into the market because they didn't have the right evidence to speak to payers to get the access and the price and the conditions they wanted. So there was about a 1-year delay from regulatory approval up to the point where the drug was made available to patients. 1 year. And that's happening more and more. Companies focus so much on regulatory that they very often forget the times have changed. As Nelson said, times they are changing. Yes, it's not like kind of 20 years ago where you had a new product, you brought it into the market, you have lots of salespeople, and then job done. Times have dramatically changed. It's much more complex to be successful. Many compounds come onto the market and then fall flat. They don't, you know, go up like this rocket, yeah, like this spaceship and get to all the patients. Very often they kind of fall down, fall flat, and are forgotten. Next slide. So if we think about physicians, payers, patients, what do they need? What kind of evidence do they need? You're all physicians, so you all know kind of what evidence you need as a treating physician. Yeah, when there's a new drug, what questions do you have? Is the drug safe? Is it effective? Yes, of course, that's because it's approved. But how do I use it? Will it be paid by the payers? Will it be paid by the NHS in the UK? Will it be paid in Germany? Under which conditions am I allowed to treat it? What is the best dosing? What happens if a side effect comes? There's lots of different questions that physicians have said are not answered by regulators. Payers the same. They are more interested in understanding, is that drug cost effective? Should we use it first line, second line, third line? Should we use it only for a restricted population? Is it actually effective in our patient population because it was only studied in the US? And then patients also have a lot of additional questions in terms of what is in it for them. Yeah. So we need to be able to answer these 3 key stakeholder groups in order to be successful with launching a new product. Next slide. Over the course of the clinical development process, there are typical patterns that we want to look into. At the beginning, and I'm not looking into very, very early, but what usually is a typical pattern in when you get to your phase 2 data is understand what will be the high-level challenges for patients, physicians, and payers. Yeah, what are the guidelines? What are the treatment patterns? And then later you need, what evidence package do we need from clinical trials, real-world data, and other sources? And how can the phase 2 data already help here? Then we come to the regulatory submission and regulatory approval, and we have questions about the HTA dossiers, the payer negotiations, and what do we need to put into publications and promotions. And then it doesn't stop there. You know, life goes on. There's other companies going for the same indication. And then questions is, how does your product compete in terms of benefits, costs versus the new drugs? Or there's payer reevaluations. There's now a whole new class of treatment, and you were part of, your treatment was part of that. And this whole class is reevaluated. And of course, you want to, you know, get as many patients benefiting from this new product as possible. But over time, how can you reach everybody? Yeah. Is it still relevant? Is it outdated? Yeah. So there's questions along the whole life cycle that we need to answer. And in all of that, statisticians are key for involvement. Next slide. So let's look into a couple of case studies. Next slide. Okay. The first question very often early on is, are physicians and payers convinced about the burden of disease? Well, if you work in oncology, yeah, or typical established therapeutic areas, then that's clear. But if you work on a compound where there hasn't been a new launch, a new product for 20 years, that's not really very much on the radar. Or maybe there's a new, there's a completely, you know, rarely known disease. What is that really the burden here? Patients do understand it, but physicians and payers very often don't understand it. Next slide. The next topic that is very often coming up through kind of about this time of the development is, what are the typical patients? Do they differ across different countries? Are patients treated differently in France and in the UK and in the US? What are the questions there? Next slide. Then when we can come to the regulatory submission, it is about how do we best communicate the benefits in a way understandable to patients, physicians, and patients. It's not so much about clinical trial summaries that we send to the FDA. We are not talking to experts. We are talking to a much more lay audience. So we need to come up with not tables, but great data visualizations, for example. And here statisticians can help you to come up with very, very nice graphics. There's a lot of development going on in this area, and you can benefit from that. Next slide. Now, there's a new competitor arriving. What do? As a statistician, you would look into indirect comparisons, into network meta-analysis, things like that, and then have valid evidence that supports you in comparing different treatments, because very often you will not have a head-to-head study. Next slide. Later on, yeah, there might be price changes. Can you then still convince the payers? What new evidence do you have? Do you have real-world evidence that shows that patients really stay very, very long on your treatment, and therefore that's very, very cost-effective? Next slide. And then later on, what are the long-term benefits of the product? With your regulatory submission, you may have only, let's say, 8-week data, maybe 1-year data. But for chronic diseases, what happens after 2, 3, 4, 5 years? What happens with patients? Are there any, you know, is a decrease in efficacy, an increase in safety problems? What's happening there? What are the long-term benefits? Next slide. And at the end, how can we convince physicians still not using the new compound? Yeah, If you think about this typical change management curve, the people that sit at the end that still hasn't embraced the changes, how can you get them to adopt this and make sure that patients in their practice also benefit from it? Next slide. So in summary, across all the different life cycle of a product, there's many, many different ways where you can work with a statistician and where you can get the best evidence available. And it's so important to work on how you communicate this evidence. Yeah? Because clinical trials and clinical trial reports are great for experts in the FDA. But when it comes to payers, physicians, patients, you need to have usually something that is a much more succinct, a really nice graphic, a really nice explanation. And you need to have more understanding, you need to have more training, because these are not experts like the people at the FDA. So that's my summary. And I'm handing back to Malcolm for the Q&A. And I think we start with a poll and see whether we have some updates in terms of the perceptions about statisticians here.
Alexander, thank you very much indeed. So, yes, as you said, we're going to go back and see if Alexander and Nelson have changed your opinions about statisticians, which, again, I'm slightly nervous about because, you know, I think it's a difficult question. So, if you'd like to push your buttons again, I think though, Alexander, I think we're going to have a slightly different result because we've got quite a few more people on the line now. So statistically, you may have a different result. So that would be interesting to see how that might work out. But pop your buttons in and let's see what everybody thinks now. Let's see if we reduce a nerdy bit.
We had 11% before for the nerdy. So let's see.
Yes. Okay. Anna, thank you. Let's see the results. That's great. So wait, well, yes, the nerdy's gone down. That's good. Difficult to understand. No, I think that's true. Smart, yes, same level. Easy to work with. Strategic. Absolutely. So, Alexander, can I ask you first what you think of that? Is that what you'd like to see? Or?
Yeah, well, the nerdy and difficult to understand went down, and the easy to work with and strategic went up. So I think
it's worked. It's worked. The whole point's good. So, Nelson, what's your view? Is that good for you?
Yeah, that's somewhat pleasing. I think we've got the Q&A ahead of us, so let's also see what other views come through that.
Yeah. I'd quit whilst you're ahead, guys. Honestly, I'd hit quit whilst you're ahead. Okay, right. Thank you very much, and Anna, thanks for doing the polls. It worked, which is always a relief for us. Right, so we got a few questions. So we've got about 15 minutes, which isn't very long because, as I always say, we want to stop on time. I've got a lot of respect for your time. You know, we want to fit this in before you start your main day's work. So, Graham, you asked the very first question. Is a 3+3 design still optimal if one's evaluating a cytotoxic? So I think I'll ask you, Nelson, first, because let Alexander get his voice back.
Yeah. So I'm going to have to say it depends, and that's maybe no surprise from a statistician. Hey, Graham. By the way, hi, Graham. Thanks for coming along today. Because what does optimal mean? I think, you know, if we reflect back to that slide that I had, the list of whatever it was, a dozen questions, what are we trying to optimize? You know, if we're trying to optimize understanding of biomarkers, that may be very different to optimizing estimation of the DLTs or, you know, the MTD. So I use the word depends because it depends what we are optimizing. And then the second thing to say is that there is a formulation of the Boyne that matches 3+3. So one can think of 3+3 as a special case of Boyne under certain circumstances. And so I would say in that situation, then no, 3+3 isn't optimal because classically it restricts you to 3 or 6 in the cohort. Whereas Boyne, you can have 1, 2, 7, 9, 3, 4, 6. You can add another one. So, you know, again, it comes back to what are you trying to optimize? I think what I like to think about is trials that have flexibility in there as well as the rigor. So it's a depends, but you can also formulate 3+3 as a special case of Boyne.
Okay. Anything to add to that, Alexander?
No, but it just highlights that it's really important to discuss about these things with a statistician, because as Nelson just said, these words like optimal, understanding really the background to that is so important to come up with a good answer.
Yeah, no, absolutely. Right, another question. Fred, good morning, Fred. Nelson, could you say a little bit more, sorry, Nelson, you're getting these questions first, my apologies. Could you say a little bit more about the choice of safety boundary in the Boyne design? Because this is too advanced for me. Is a plus or minus 40% widely used, or is the scope to vary this, for example, to account for anticipated high risk of escalation? There's a second part of the question, but I'll let you do that bit first.
Okay, yeah. Thanks, Fred, for the question and also for joining. So yes, there is flexibility around the plus or minus 40%. The authors have shown theoretically why 40% is a good default, but no, you can change that to, and in your case, take the context of potentially being a bit more conservative on your escalation. Another thing that the authors highlight is, but you probably don't want to make that sweet spot too narrow. And this is partly because, the small sample sizes, as you saw from my worked example, it's 1 out of 3, 2 out of 3, 3 out of 3. Those percentages, you can only get 33%, 66%, or 100%. So if you make it too narrow, that sweet spot, then you really can't optimize when you're escalating and de-escalating. And the authors give some guidance that plus or minus 15% is about as narrow as you want to do in general. But, and I'll plug it one more time, but doing simulations for the context that your team is in will help you fine tune those. But yeah, there's flexibility. You can use simulations to fine tune what they should be, subject to the author's recommendations. Don't make them too narrow.
Okay, thanks, Nelson. Alexander, any further comment on that one?
Nope. Okay,
fine, good. Right, Claire, good morning, Claire. Lovely seeing you here, thank you. So this is quite a long question. So Alexander, I'll give this to you to start with because poor Nelson's been inundated at the moment. When designing a clinical development plan for agents that are still in preclinical development, often for potential investors, I know that one very well, it's often necessary to provide trial outlines, estimated patient numbers and timelines long before much is known about the drug. You know that one too, Claire. And before involvement of a statistician. Yep, absolutely. Is it reasonable to use a 3+3 design for these estimates and to say that more sophisticated trial designs will be used and are likely to result in lower numbers? Or do they sometimes result in higher numbers? And I think that's quite a true one, Claire, isn't it? Because I go and, I go as a standard, the lowest common denominator, and because they want something, and they get a bit nervous if you start chucking different things at them. So, Alexander, what do you think? Is it, it's a business question as well, isn't it, really?
Yeah, but you need to be pragmatic here. Yeah. So, yes, it can result in higher numbers as well. Yeah, so it's not necessarily that better methods are always smaller, because it, as Nelson earlier said, it depends on what is better. Yeah, what is optimal. Yeah, if your criteria for optimality changes as you learn more, yeah, you might have different assumptions, different trial designs, and therefore then also different numbers. But it's you need to come with some kind of ballpark, yeah. And I think people appreciate that things will change over time. Yeah, I think it's more important to communicate that yes, that is our first guess, and there will be some changes upwards and downwards.
Nelson, anything further about that part? I'm sure there is.
Yeah, I certainly echo what Alexander said. I think it's maybe not a surprise. I'm going to say, well, the price of not involving a statistician earlier on is potentially a weaker proposal to the investors and stakeholders. And I'm sure a number of you have. I mean, I was on an investor call, and I was being grilled by the Wall Street investors for one biotech company. Really, really smart questions on statistics in here. Now, that's just, one recent experience of that. I would like to think that over time, some of these investors are saying, well, hang on a minute, sample size is not the only thing. Why are you proposing this design that is highly likely to choose the wrong dose? And we're going to take that dose forward into bigger studies, and we're going to throw money on the wrong dose. So I have to acknowledge and recognize statisticians don't always get brought in earlier, but I would like to encourage people in this kind of forum and their colleagues is like, look, reach out to a statistician. It may be with a small number of hours work, an option which is more flexible and has got better properties in terms of choosing maybe the maximum tolerated dose will go down well with investors, and particularly those who are really very savvy about statistics.
Yes, it's the same actually later. Yeah. I just brought this example where people thought, oh yeah, our evidence is sufficient, but no, that might not be. And statisticians can look into data very, very differently. Yeah. So when we speak about data, physicians very often think in terms of means and summary statistics. Statisticians think in terms of the patient level data and what we can also get from there additionally. Yeah. And they may think about lots of different data sources, not just the clinical trial at hand.
Claire, does that answer the question adequately? I can't hear you, but that's okay. You're nodding. All
yes,
right.
Thank you. It's just that sometimes companies don't give you say, oh, I'd like to involve a statistician, and they say, oh yes, later, or something like that. Yes, thank you very much. That does confirm my suspicion that sometimes the sophisticated trial designs result in higher sample sizes, which often with these small companies fills them with fear. But yes, you for a better trial design and more robust answers. Thank you.
Okay, Claire, thank you. And Jose, good morning, Jose. How to optimize dose escalation decisions for immuno-oncologic agents where their drug takes some time to start the action? Right.
Should I take a crack at that first? Or
Alexander?
Do you want to go, Alexander?
So how to optimize dose escalation? Okay.
So just really briefly, there are extensions of a number of the designs that I've presented today, which will have, for example, so-called time to event or delayed event modeling incorporated into it. So for example, if the length of the DLT period in order to do the first escalation is very short, maybe 1 or 2 cycles, you can then build in later events into your study design and modeling. These designs do rely on assumptions. So, like any model, if those assumptions hold, great, it's an improved design. But the short answer is yes, there are extensions, and the MD Anderson is one place you could start poking around and having a look at some of theirs, some of those, or we can connect afterwards if necessary, Jose.
Okay, Alexander, anything to add to that one? Or...
No, but I could shortly comment on the comment from Rudy about key is to have organized your data in an agile way. Yes, that is absolutely important because unlike earlier in the clinical development area, when it comes to launching and commercialization of products, you have many more internal stakeholders. Yeah, you can have hundreds or even thousands of people that work with your results and your data. And also externally, it's much bigger. So making sure that you have a easy to access the data, both in terms of the results and the underlying patient level data is important. In addition, you also need not just the data, but you need explanations about the data. So having a platform where you have frequently asked questions where people can understand, okay, why is our design different than the one from the competition? What are the strengths and limitations with this data? What claim can I do with this data? Or I want to say this claim, and what kind of data do I need for this? Is there maybe the results that are not yet available and that could be made available? So you need to have processes for managing all these kind of interactions in a good way. And actually, the bigger the company, the more challenging this becomes.
No, it's very true. It's very true.
Malcolm, if it's okay, I have a question that someone would just like to ask directly, if that's okay.
Yes, let's go ahead.
Hi, Nelson. Hi, Julie. Alexander, thank you for a very nice presentation. I have a question, and I will try my best to actually ask for it. You know, with either of these design 3+3 or BOIN or dose escalation with overdose control, you know, lately, let's say in last 3 years, what we have seen some serious AE-related concerns, or sometimes probably death in either expansion cohort. This is mainly in context to oncology studies, or we may see the death in the phase 2 study. And being part of about more than 20 early development studies now in last 6 to 7 years, what I tend to see, there is some element of luck with escalating dose using 3 patients, and that's partly, in my experience, what I see. Sometimes you have the patient with the low disease burden, a good performance status, not heavily pre-treated, and when something goes, you know, when something passes the hurdle, it goes to the next stage, and suddenly there is a less scrutiny in terms of targeted eligibility. You have the patient with more disease burden, and bang, either grade 4, grade 5, and or probably death. You know, how can we eliminate some of the uncertainty with escalating on the 3 patients data? Sorry if that makes sense, but I think it's the best to ask directly rather than type in the chat box here.
Malcolm, I know we're coming up on time.
Yeah,
I have a very quick answer, but you probably want to close out, do you? Or
no, we got about 30 seconds, so it's okay.
Yeah, thanks, Sweetang, and we can talk about this at length. I think what I would say is that, you know, selecting a design that allows you flexible number of patients in a cohort allows you, for example, as you're approaching MTD, you can maybe recruit 6, 7, 8, 10, 12 before you do a full expansion. So I think that's one thing that can actually help, is choose designs that have flexible sample size and gate the full expansion by maybe taking steps towards there. Just a couple of ideas that come to mind.
Okay,
thank you.
Thank you very much indeed. Alexander, anything to quickly add to that, or?
Oh, thanks so much.
Okay, fantastic. Look, just to me to say thank you very much to Nelson, Alexander. Can't thank you enough. Really fascinating. I think we've proven that statisticians are not nerdy. Well, they can be, but generally speaking, you guys are not, in terms of being so approachable and actually informative about the way you put forward statistics. So thank you. Thank you to everybody who turned up today. That's fantastic to have you here. We're going to do another one relatively shortly, in a couple of months' time, we hope. My thanks to Anna for setting this up, to Liz and the rest of the team for doing it as well. Have a fantastic day. Any points that you want to bring to us in terms of putting forward to Nelson or Alexander? I'm sure they'd be more than happy to answer them. And just finally, we got CPD points for this. So if you'd like to approach us, if you need CPD points, if you'd like to have a piece of paper or something for them, approach us and we will send that form over to you. Okay, thank you very much. Have a great day. And my thanks again to Nelson and Alexander. Thank you.
Big kudos to you and Anna and Liz as well for organising and hosting.
Pleasure. Absolute pleasure. Right. Thank you.